for joining me on my ride. That was a revisit to episode 19 and episode 10. Episode 19 was Nick Steinmetz's paper. Episode 10 was Ann Churchland's paper. Those two papers were lambasted by me for not having a suitable hypothesis and not testing anything really in their papers. This is not to take away from the fact that they're excellent observations about how distributed networks are arranged in the cortex. It's not to say that we didn't need those measurements. But Lydia is on to something here when she says that neuroscience is often missing a hypothesis and often missing the replicable data that would be necessary to sort of impart an understanding on the brain. She's not wrong there. The problem is that right now we're at a stage in neuroscience where we have a lot of technology that can get us a lot of data, but we don't have suitable models and suitable theories to explain the data that we're collecting. So in the meantime, we're left with just making massive observations and trying to explain them as best we can with descriptive statistics and correlative measures. Now, you can say all you want about using the semantics correctly and not calling it encoding when it's not encoding. Just saying correlation might make Lydia a little happier. But I've seen other examples of Lydia on the internet where she just goes too far. The best example I have is when she's attacked Steve Ramirez. I'm not really sure she even understands what the Tonigawa lab does and what Steve Ramirez lab is doing with their molecular techniques to tag cells and to control them during behavior and during learning. And that leads her to believe that there's some fraud or something going on. She actually accused Steve Ramirez of fraud on PubPeer. And I think this is where she starts to cross the line. Essentially, you break down any bridges and stop the discussion the moment you accuse people of being disingenuous. And I honestly believe that neither Nick nor Ann nor Steve are trying to be disingenuous about their data. They have these huge observations, they have these huge data sets, and they want to describe them beyond just, here, have it. That's what Nick tries to do. That's why figure 4 and figure 5 are interesting and novel observations. But what can we learn from these observations? Not a lot. And the reason why is because the paper that comes before it also doesn't really manipulate the experience of the animal. Yes, they have a neurometric model that can predict the outcome after optogenetic inactivation, but let's face it, if I have a ball in a box and it's half black and half white, and I put it inside of the cover of the box and spin it around, I can call that a pretty adequate model for the description of day and night. But it actually doesn't belie a lot of understanding as to the generation of that pattern. The same could be said for this neurometric model. I don't think Nick thinks that the left and the right side compete with one another for adversive or promoting behavior. I don't think he really believes that the brain is using his model to solve the problem, but his model is capable of predicting and explaining the results of optogenetic inhibition. But we have to be careful to jump from there to labeling it as encoding, labeling it as understanding. That's not what it is. It is an observation. It's a good observation, a powerful observation that still has yet to be put into a functional context that makes us progress farther down the road of understanding how the brain works. I then gave you two examples of papers in the retrosplenal cortex that came out very recently. One from Cardiff in the UK, where they basically do only post hoc analysis, waving gratings, running on a wheel, and seeing if these two stimuli add up in the retrosplenal cortex. In light of Mike Hasselmo's recent paper, in light of the 2015 paper from Nitz and Alexander that is cited in that paper, essentially we haven't learned anything from this retrosplenal paper. On the other hand, look at Mark Harnett's recent paper in eLife. There he does at least 2, if not 3 manipulations to demonstrate that his interpretation of the activity in retrosplenal cortex is likely correct. And then I let you in on Adam Hatman and Jackie Schiller's tour de force that's now currently on bioRxiv, where they dissect output signals in layer 2-3 of M1. That paper shows you how to look at large quantities of data and then manipulate the experiment and look at how those large quantities of data change. Do they still know a lot about how M1 works with the striatum or with anything else to generate behavior? No, they don't. but they have one small piece of the puzzle that through a series of manipulations I definitely believe. So you got 5 papers or 6 papers to look at, Nick's paper and paper, 2 papers from retrospinal cortex, and then a tour de force from the Hantman-Schiller lab combo in bioRxiv. I hope you enjoy it. If you liked it, like and subscribe. The ride's coming soon. I'm talking till the very end because I cover all of this as best I can. I hope you enjoy it. See you next time. Pittsburgh and Universe. This is JC on a bike. My name is JC and this is Journal Club on a bike. I'm getting started quick tonight because I got a lot to cover. I'm going to try and revisit episodes number 10 and episodes number 19 here and try to push this this discussion forward. And I'm pushing it forward in the context of a Twitter personality, the first name of Lydia. Lydia is a negative force to be reckoned with on Twitter, and a very anti-modern neuroscience lady who has a lot of problem with p-hacking and post-hoc correlation, and feels like the use of these things in neuroscience is so rampant that neuroscience is basically nothing anymore and has become pseudoscience. Now, I'm going to argue that one of the reasons why so many people are blocking Lydia is because she's become irrational. And it's not to say that some of her concerns are not valid, but most of our concerns are amazingly semantic, and they're real concerns. But the sort of aggression and accusing people of fraud is not necessarily the way to make friends and to make progress with an intellectual argument. And so what I'm trying to do is put a little, let's say, fresher meat on those bones so that we're not just, citing the same 3 papers over and over again and saying that neuroscience sucks, because that's not going to get us anywhere. And so first of all, I just want to say that I think that she has an objection that is somewhat valid. And that objection stems from the widespread use of the coding metaphor in neuroscience. And that coding metaphor simply means that if you see action potentials in neurons around a arbitrarily selected time point in the experience of the animal, then that correlation can be called, in neuroscience, you could call that encoding. Encoding of whatever happens at that time that you deem relevant. And that's what she's frustrated with. And in episode 10, we look at Ann Churchland's paper where single trial dynamics are dominated by movement. This paper showed absolutely nothing new and taught us nothing new about how the brain works. Nothing at all. And of course this frustrated Lydia, because even the title is kind of dumb. The title actually says we don't understand anything about our data. That's what the title says. Single trial variability is something about movement. I mean, it's literally, we can't explain our data except through the movement of the animal. And that's crazy. That's not a scientific conclusion. That paper looked at a task and looked at landmarks in the task, tried to use a linear model to explain the activity observed in the brain, at various resolutions from bad to decent, and showed that their linear models only really worked well when the linear model was based on the moving pixels of the video. So this is a gigantic paper. It gets touted around, and she goes around the United States giving presentations about this data, probably invited talks that are paid for. And it's not science. I'm sorry, but it's not. It's an observation. It's a beautiful observation. We need to make more observations. And the reason why we need to make more observations is because we don't have a working theory of how the brain works right now. And Nick Steinmetz would argue that the reason why we don't have a working theory is because we've always been recording from just one place at a time. And that's why Nick Steinmetz is so excited about his paper from Ken Harris's lab, where distributed coding of choice and engagement or something like that in the cortex. Again, we need to make these observations. We need to have this data. We need to see it. And admittedly, I sold Nick's paper a little bit short. I stopped on figure 3, kind of making fun of the figure legends being like, visual cortex information shows up in visual cortex first. And there are 2 pretty neat observations in this paper. The first neat observation is in figure 4 of Nick's paper, where they show that correlative firing, correlative signals to choice show up in the cortex, in the basal ganglia, and where else? And the midbrain, I think. It's the basal ganglia and the midbrain. And the cortex. Choice shows up there simultaneously. That's in figure 4. That's a neat observation. No one had observed that before except for an fMRI. fMRI has shown us that there's lots of simultaneous signals in the brain, but not at the single neuron resolution level like what Nick has shown. Another really special observation that he makes is that there is a brain state that corresponds to a lowering of cortical activity, an increase of subcortical activity that correlates well with or corresponds to their ability to discern low contrast stimuli. No one has ever observed this sort of brain state change before. But the problem with this paper is that Nick and Ken Harris's lab haven't done anything to show us via manipulation or experimental test to show us that any of their data is real. It's just correlative. It's all analyzed after the experiment. And one of the reasons why I was frustrated with that data is also because in that paper they don't really describe the task or the neurometric model. They just refer to another manuscript, which is available and I have read, and I can get so far in it, but not everywhere. Can't read all the papers every night. But I understand the gist of it now, that they have this model and it can explain the effects of optogenetic inhibition, yada yada yada. But again, the model's explanation and the model's predictions, it's not a very big gain of understanding there. Do they really believe that one side of the brain is, fighting the other side of the brain to decide whether to choose left or right? Because that would be the implications of their model. That one side is controversive one way and promoting the other way, and then vice versa. Like, is that really what they think? That's their model of brain function and choice making? That can't be. I might have that wrong, But I'm just trying to make a general point here that even with the two papers and even with that model, I mean, what insight are we gaining besides the observation? We're not really gaining anything because, again, you're not claiming that your model is an accurate representation of the calculation in the brain, are you? You're actually just claiming that your model works. I mean, if I put a half black and half white ball into a box and spin it around, that's a model for a day and night cycle. But it doesn't belie that I understand how the day and night cycle works. And so if you make a neurometric model that explains the effects of optogenetics on your simple system, that doesn't necessarily mean that your simple system explains the brain. It's just a way for you to explain your data and the patterns that you see within it. And that's, again, the frustration with using the word encoding. So I've got two examples that I want you to look at and read. I'm just going to cover very briefly, and then I'm going to cover one really big one really hard. So there are two papers that recently came out in the retrospinal cortex. One is in eLife by Mark Harnett's lab, and the other one is in cerebral cortex, and this one is from the Ranson lab, I believe, in the UK. So the Ranson lab has put out what is basically, we're going to show visual gratings to the retrospinal cortex, and we're going to look for cells that respond to the gratings, and then we're going to run them on wheels and see if the wheels and the gratings combine in retrospinal cortex. Now, in light of the fact that we just saw a paper in Science showing boundary vector cells, egocentric boundary vector coding in retrospinal cortex, which cited a paper from 2015, thank you. He just cited a paper from 2015 that also showed, thank you very much, you don't have to get out of my way though, that also showed this in vivo, that these were combining motion and vision. So to do it, this paper on a wheel, and then do no manipulations at all, but just do post hoc correlations to find the cells that match your model. I'm sorry. This paper would have been great if it came out 5 or 6 years ago, but now it's just baloney. So I don't want to insult you guys, but man, we're not gaining anything at all from this paper. And I'll show you why. If you look at Mark Harnett's paper, he uses exactly the same prep. It's an animal on a wheel. He thinks that the retrospinal cortex encodes something visual, but something concrete, like landmarks. And then he does experiments to manipulate the animal's experience to test whether the retrospinal cortex is signaling landmarks. It's a really nice paper because the animal runs in virtual reality. There are two landmarks that provide different locations for the reward. So when they see one landmark, they should know they have to run farther than for the other landmark before they get their reward. And the animals learn this task, and then they mess with the relationship between the wheel turning and the landmark movement. This is manipulation. This is an experiment with a hypothesis. And if you want to see a real tour de force of recording from lots of neurons and not necessarily knowing what they all do, but still learning something about the brain, Adam Hatman and Jackie Schiller have just put out a paper on bioRxiv that's just a frickin' monster. They're going to look at outcome signals in M1, and specifically they're looking at two different populations. They're looking at layer 2-3 pyramidal cells, and they're looking at a subset of layer 5 pyramidal cells called PT pyramidal cells. And by looking at these two populations separately, they're going to try and look for outcome signals that tell the medial entorhinal cortex, that tell the they're looking for signals that tell the motor cortex what happened on the last trial. Did you miss the shot? Did you make the shot? And in this case, did you grab the pellet in 2 or 3 tries, or did you try 3 or 4 times and miss every time? And so it's not a perfect task. They don't get it right every time. They make mistakes, and that's what allows them to find this error signal. And so in figure 1, they show you the signals, and they show you that they get a little bit more signaling from the misses than the makes because they try harder on the misses. And then figure 2 shows the outcome signals in layer 2, 3 versus layer 5. And you see that there's really no difference between positive and negative outcomes, success and failure in layer 5, but there is a difference in layer 2, 3. Certain cells signal success and certain cells signal failure. And more importantly, in figure 2, they show you that this develops over training, and that's pretty cool. So now they have this outcome signal in layer 2, 3, and they want to see how it really works. So in figure 3, I think they're going to make a linear model already, and then line up the layer 5 and the layer 2, 3 cells to different behavioral experience landmarks, like, you know, when they grab, when they supinate, when they get it to their mouth. And when they do that, what they see is with a generalized linear model, and you line it up like peri-action and post-action, then most of the activity in layer 5 can be explained with a generalized linear model as long as you put this activity around the action, around the movement. But layer 2, 3 doesn't. Layer 2, 3 is much easier to explain if you look at post-action time points and you use outcome. And so this is just a post hoc analysis that tries to support the observation they make in figure 2. Figure 4, I believe, counting figures in my head. Figure 4 is going to do principal component analysis to try and look at the population activity in layer 2, 3 versus layer 5 and see if they can see a trajectory difference in the principal component analysis. Nice dog! And that principal component analysis immediately reveals 2 different trajectories in layer 2, 3 depending on success or failure. And there are 2 different trajectories in layer 5, but they're much more similar. And so statistically speaking, there's a giant difference between success and failure in layer 2, 3, and very little difference in layer 5. So that's really neat. What they also find in this figure is that the starting state of the layer 5 neurons is dependent on the trial before it. And so this gives them a really interesting clue that they're going to work on later. The next thing they do, I think this is figure 5, is that they force the animal, or make the animal stop reaching and instead use his tongue to get the pellet. And what this does, predictably, if you think their hypothesis is correct, that the air signal in the motor region they were looking in goes away because the tongue is represented somewhere else. That's a really cool result. Then in figure 6, they show that it's not the eating, it's not the fact that they get the reward that signals the difference between success and failure. And that you might have thought, right? They're getting sucrose nuggets. So when they get the sugar, then 2, 3 signals. And when they don't get the sugar, 2-3 signals. So of course these people have a really spectacular control here. They use 3D printed sucrose pellets that aren't made of sucrose but made of plastic, and the animal performs the task similarly and even puts them in his mouth and then has to get it out, you know, like yuck. But still, the success and failure signals are present in layer 2-3, despite the fact that they're now getting plastic pellets. That's pretty badass. That's really cool. And again, he's making massive recordings from massive numbers of neurons and trying to find patterns in them that are meaningful. But much more importantly, they are able to tweak those patterns or tweak the experience and make patterns and have the patterns respond in predictable ways. So 6 is plastic pellets. Sorry about that. 6 is plastic pellets. Great result. I'll just start from the beginning again. So just to summarize where we are so far, figure 1 is the basic recording. Figure 2, outcome signals in 2-3 and not in 5. Figure 3 is a linear model to show that motion describes layer 5 and outcome after the action describes layer 3. Figure 4 is PCA analysis, which shows the trajectory of 2-3 splits for success and failure, and 5 doesn't really. 5 shows that the success and failure rate signals go away when they use their tongue. 6 shows that with plastic pellets there's still a signal, so it's not reward. And then 7 takes advantage of this thing that I pointed out in figure 4, that the start state of the layer 5 network is dependent on the outcome of the previous trial. So if they optically inhibit layer 2-3, during this time when the signal for success or failure would be exhibited, then they see that the start state of layer 5 does not reset. I mean, come on guys. Come on Nick. Come on Anne. That's how science is done. You make some observations, and then you make a hypothesis about those observations, and then you perform an experiment where you make predictions about the outcome based on your hypothesis. And then after the experiment, you reject or accept your hypothesis based on the outcome of the experiment. Now keep in mind, I'm not arguing that we don't need these observations. But I am arguing that Ann Churchland and Nick Steinmetz don't know any more about how the brain works, or don't know a lot more than they did before their papers came out. Especially Ann Churchland's lab. That paper is, I'm sorry, it's scientifically vapid. It is just observations, and that's fine. But I just can't understand how people are still allowed to use linear models to describe a system that nobody in the world thinks is linear. And I'm still really surprised that people still think that action potentials in most of the brain can be thought of to spike and have information encoded in those bits that is as simple to decode as to look at what the animal is doing from your perspective as an experimenter at any given time. That's absurd, given the fact that the vast majority of the variation in any of these experiments, as Ann Churchland so wonderfully showed us, can be explained simply by a linear model which transforms the edge of the animal in a video to an equation. I mean, are you kidding me? Are we done now? We can just stop?